Hi everyone, welcome to my presentation today about bioinformatics. So essentially, I wanted to make a little bit of an introduction as to who am I and who my mentor is. So my name is Medhasree Allu and I'm currently calling from Dallas, Texas. My mentor's name is Joshua Price. He's a mentor with a valuable background of experiences in fielding in biotechnology and bioinformatics with genetics and lab experience. Now, why do I choose to research this topic? My interest may be a bare and generic response, but the heartfelt truth is truly what drives me towards bioinformatics and research through experiments of genetics to hopefully produce cures and interesting discoveries that can also benefit those who suffer. My family additionally has a huge history of CVD and genetic diseases, which more than a burden, in my opinion, acts as a lending base to help me understand and comprehend various aspects of genetic diseases, but also the various protein processes and genes that mimic a various gene PCSK9, which plays a key role in cardiovascular disease. I truly believe the passion comes from our current status quo of research, but also experience, which blended together makes a true desire for answers. So the question we're going to be focusing on today for my presentation is, are there other genes whose expression levels predict LDL levels, and are these genes capable of interacting with the gene PCSK9? This is a little table of contents of what we're going to be reviewing today. So LDL, CVD, PCSK9, project, scRNA, and various genes. So, what are LDL levels? LDL levels are the levels of cholesterol, and they mainly show the direct correlation to heart disease or stroke. When having high LDL levels, a huge risk can occur with the high amount of PCSK9 gene with a high amount of the bad cholesterol, leading to a massive exposure of CVD and health obstacles from the lack of heart support. Good LDL levels contain a good amount of cholesterol levels, which show the great amount of cholesterol needed, which also express the good regulation of heart health factors such as obesity, lack of proper eating habits, and more, which can cause LDL levels to spike, showing the negative side effects. So essentially, if you have low LDL levels, this can cause a less risk of CVD due to the restrictions of plaque buildup. But if you have high LDL levels, this will cause a high risk of CVD, cardiovascular disease, due to the implementations of plaque buildup. Now, what is cardiovascular disease? It's not only a disease, but it is an unfortunate lifestyle. It consists of unhealthy eating habits, non-sufficient sugar levels, smoking habits, and various other genetic reasons which play a role. Essentially, plaque and arteries restrict proper blood flow through the ventricles and eventually the heart and brain, causing a deathly heart attack. So, what is PCSK9? PCSK9 is a very important protein. It's made by the liver to control how much bad cholesterol stays in your blood. So, how does PCSK9 do this? It binds to the LDL receptors, which are found on the liver cells. PCSK9 binds to the receptors, destroys them, and ensures that they won't use the LDL cholesterol. It's essentially a code book destroying and restoring the receptors that can harm or generally affect the process or pathway of the bad cholesterol, leading to serious heart obstacles. PCSK9 is widely reviewed, and many scientists and researchers throughout the world use this gene to formulate experiments and receive answers. For example, one study I read about was an animal study with mice through lower cholesterol without the gene or the regulation of PCSK9 having less risk of CVD. Other experiments showcase this as well. A note is PCSK9 is not inherently good or bad. It's just a gene that regulates cholesterol levels. However, when there's too much PCSK9, it can raise the bad cholesterol by reducing the number of receptors, and it will eventually lead to harmful side factors. So in my project, I'm currently identifying samples of human heart, blood, and liver samples through available data sets in the internet, which show the potential experiments that can lead me to my conclusion findings regarding other genes. By using statements such as single-cell RNA sequencing, liver high and low LDL levels, I'm able to find sites with numerous reviews. With these available pieces of procedures, it is available for me to formulate my own conclusions about a diversification of genes. Now, what is scRNA? scRNA is called single-cell RNA sequencing. This process is very important and powerful because it is obviously used throughout the entire scientific field today, and it essentially shows which genes are present or not in an individual cell. It mentions and evaluates the gene expression in a very thorough and well-mannered process, expressing the findings through technology, labs, and more. scRNA is very important due to the fact that it reveals and shows various developments of the genes in the cells through complex evaluations, helping in cancer, neurology, and CVD as well. Through the stages of examining, extracting, and sequencing and data analysis, scRNA clearly examines the individual cell composure of a certain gene exposure. So these are some of the various genes that I have experimented and researched with that essentially interact with PCSK9 and have a role with LDL levels. We first have LDLR, which provides instructions for making the LDL receptor. It's a gene that makes the receptor, removes the bad cholesterol from the blood by bringing it into the liver cells. PCSK9 then attaches to the receptors, causing them to break down, reducing their number. When you have fewer receptors available, less cholesterol is cleared, leading to higher LDL levels. So the amount of LDLR the body produces directly affects the cholesterol levels or LDL levels and how much influence PCSK9 has. APOB produces APOB 100 protein, which is a main structural protein of LDL particles. It helps with the bad cholesterol by attaching to the LDL receptors so it can be cleared from the blood. If there are mutations in APOB, LDL cannot bind properly and cholesterol builds up. While APOB doesn't directly interact with PCSK9, it affects how much LDL is present. And more LDL means more pressure on LDL receptors and greater activity from PCSK9. The last one is LDLR-AP1. So this gene essentially helps the LDL receptor function correctly by producing an adapter protein that facilitates the internalization of the LDL receptor complex into the cell. Mutations cause a severe form of high cholesterol. So essentially, it's a gene that helps with the LDL receptors work properly by allowing them to pull LDL cholesterol into liver cells. If this gene doesn't function properly, the receptors cannot be removed and LDL will be less effective, leading to higher cholesterol. Although this gene doesn't directly interact with PCSK9, it plays a key role in the same pathway and influences how well PCSK9 and LDL receptors work together to manage cholesterol levels. So a little bit of a more debrief into these genes. Some genes that affect LDL directly influence PCSK9 because they work in the same pathway. So the genes that I mentioned in these bullet points are the genes that work in the same pathway. So LDLR, like we mentioned, is PCSK9's main partner. PCSK9 controls how many LDLR proteins are on the liver surface. If LDLR levels change, PCSK9's effect also changes, showing the direct interaction between both of the genes. APOB, this essentially makes the main protein on LDL particles, doesn't directly bind to PCSK9 unlike LDLR, but it influences how much LDL is around for LDLR to regulate. So APOB essentially affects LDLR, which affects PCSK9. APOE helps clear LDL from the blood. Its effect depends on LDLR, so it indirectly ties to PCSK9 as well. Other genes affect LDL but indirectly interact with PCSK9. For example, HMGCR, which essentially is how the liver makes control and changes the LDL levels, but they don't influence PCSK9 directly. Essentially, some influence directly and some influence indirectly. Finally, in my conclusion, the usage of PCSK9 in studies shows a broad advantage to future experiments and studies, showing the importance and significance of the gene to CVD, leading to the experiments of other genes interacting and contributing to the LDL levels of PCSK9. Thank you so much.
Thank you so much for that presentation and for sharing more about what you researched. As a reminder, if anyone has any questions, feel free to add those to the Q&A section. It might be under the More tab in your Zoom. But in the meantime, I was kind of curious, where do you think this project goes next? Are there any specific genes you'd want to explore more and follow up on? Or yeah, what do you think are kind of the next steps for this project?
So some of the genes that I researched I obviously implemented into my presentation. There are other genes that I would definitely like to dive deeper into because this is like a really good passion of mine, and it's really nice to research more because there's truly a lot more information we don't know about these genes. So yeah, I would like to see this presentation making maybe like some sort of invention or some sort of discovery into like genes and bioinformatics.
Very cool. Do you also see maybe a connection between any other kind of disorders that some of these genes might have?
Yes, there's another disease called familial hyposcorsy. It's like another disease, which essentially, if there's a mutation in LDLR, which is one of the genes that works directly with PCSK9, it eventually leads to the disease FH, which is the familial disease. So yeah, there's truly like there's other diseases that also come with mutations from the genes. So CVD or cardiovascular disease is just the broad disease that's focused on.
Great. Thank you so much for sharing.